Hereby, I open this academic ceremony in which Yanchao Zhang will defend the academic thesis expanding the genetic toolbox for engineering Commensal Clostridia as live biotherapeutics. May I invite you to present the summary of your study and the conclusions of your thesis.
Dear Prorector, Committee of Appointment, colleagues, family, and friends, thank you for being here today. I'm honored to present the research I have conducted for my PhD dissertation. Throughout this presentation, I will walk you through the key elements of my work, starting with the background and motivation behind my research, followed by the methodology employed, the result I have obtained, and finally the conclusion these findings make to the field. Before we dive into the details, I would like to take a moment to thank my supervisor team and assessment committee for their guidance, support, and valuable feedback throughout this journey. I also want to extend my gratitude to my colleagues, family, and friends for their encouragement and understanding during this process. Let's begin by exploring the background behind this research. Firstly, live biotherapeutics are dynamic and distinct from traditional drugs, employing human cells and microbes for disease treatment. Among microbes, Clostridium is a genus of spore-forming, strictly anaerobic and gram-positive bacteria. They not only play an important role in various industrial production of biochemicals and biofuels, but also themselves inhabit the intestinal tracts of animals, including humans, and then play a crucial role in host health. Actually, Clostridium as live biotherapeutics has a historical research, especially in cancer therapy. Studies and trials have shown that via intravenous injection, Clostridium spores can colonize into the tumor due to the hypoxia environment inside, and then lead to cancer cell death. To improve cancer therapy using Clostridium, two strategies called Clostridium-directed enzyme project therapy and Clostridium-mediated immunotherapy were proposed and are being investigated via expressing project-converting enzymes and molecular components from immunotherapy, respectively. And some Clostridium species themselves are probiotics, which can also be promising vehicle for various gastrointestinal diseases, like inflammatory bowel disease and passenger infections. Overall, utilizing Clostridium as a promising vehicle for drug delivery presents a novel avenue for disease treatment. In order to achieve these goals, this thesis expands the genetic toolbox for efficient engineering of Clostridium sporogenes and Clostridium botulicum as live biotherapeutics for treatment of cancer and GI diseases. As shown in this figure, Clostridium can be reprogrammed to produce drugs by using different promoters for variable drug doses and efficient genomic engineering tools for rewriting chromosomes as we want. In addition, there are some requirements for live biotherapeutics with incorporation of biocontainment elements for biosafety, regulatory, and a visible reporter for effective tracking of engineered Clostridium cells. To generate a recombinant Clostridium, plasmid is a common DNA vector used widely for drug production. They occur naturally in various bacteria with concerns of transferring antibiotic-resistant or virulence elements. Besides, scientists have used plasmid to produce human proteins, like insulin for diabetics. In this thesis, we exploit plasmid to achieve drug production in recombinant Clostridium. As shown in this figure about the cloning process, by cutting and pasting, DNA for drug production can be inserted into cloning plasmid, resulting in recombinant plasmid. The plasmid would be transferred into equally cloning streams for further DNA sequencing. Then, the verified recombinant plasmid would be further transferred into Clostridium, resulting in recombinant Clostridium-producing drugs. Regarding the details of recombinant plasmid, we need a replicon from Clostridium, enabling plasmid replication within Clostridium cells for drug production. The plasmid needs another different replicon from E. coli, allowing plasmid replication during cloning process using E. coli cloning streams. During cloning process, both antibiotic-resistant marker and cloning sites are also required. Regarding the cloning site, classic cloning process using restriction enzymes has been widely used for Clostridium research, which needs multiple steps and might waste the amount of resources. While Golden Gate assembling was recently reported with one-step cutting paste for plasmid cloning, by which 50+ fragments can be efficiently assembled within 5 hours. As you may see, compared to classic cloning process, Golden Gate assembling can clone plasmid without including restriction enzyme sites, meaning scarless with no extra DNA adjunctions, which could be important for controlling drug production in Clostridium. Therefore, new series of Golden Gate-based cloning plasmid were generated in this thesis, enabling us to have scarless, rapid, and efficient cloning process for plasmid generation. During generation of recombinant plasmid in this thesis, we were faced that some drugs are challenging to be cloned and produced. Briefly, plasmid sequencing results show frequently incorrect sequence of drug's DNA, including DNA substitution, deletion, and insertion. Because all use promoter in this thesis were validated with good performance in both Clostridium and E. coli. The drug would be consistently overexpressed in E. coli when generation of recombinant plasmids. We hypothesized that this unnecessary drug production in E. coli during cloning process would be toxic to E. coli, resulting in the observation of deactivating production of correct drugs. This challenge highlights the need of independent gene control between E. coli and Clostridium to drug production only in recombinant Clostridium. In order to solve cloning issues, we generated libraries for gene regulation in this thesis, especially in the validation of start codon. We find that E. coli is sensitive to start codon change, but Clostridium is not. Start codon is required for every gene production to proteins, and is recognized by ribosome, then initiates translation process to proteins. Usually, AUG is commonly used within bacterial genomics. But previously, other group reported another 6 non-AUG start codon within 69 bacterial genomics. Because of our curiosity, we validated this non-AUG start codon within E. coli and Clostridium. Surprisingly, compared to AUG start codon, 50 to 100% drug production was observed in Clostridium, while only 0 to 5% drug production in E. coli when we use these non-AUG start codons, suggesting the existence of different translation initiation between E. coli and Clostridium. This finding offers us an opportunity to independently switch off the drug production in E. coli, bypassing the potential drug toxicity during cloning process. For example, using libraries of gene regulation in this thesis, we successfully express a SECB gene from Bacillus subtilis, which can convert sucrose into leavens, resulting in cell death in bacteria. This gene has been widely used as sucrose-sensitive marker in bacteria instead of Clostridium. These gene production issues were overcome by introducing strong promoter in Clostridium and employing one of non-AUG start codon to independently switch off gene production in E. coli, permitting the creation of sucrose-sensitive Clostridium butyricin. Therefore, non-AUG start codon enable us to easily cloning of toxic products in this thesis. Here, we can efficiently generate Clostridium with recombinant plasmid for drug production. However, using recombinant plasmid with antibiotic-resistant marker has a risk of spreading antibiotic resistance in the environment and the requirement of plasmid maintenance relying on antibiotics. Therefore, we investigated chromosome integrating for stable drug production free from antibiotics using well-known CRISPR-Cas technique. Briefly, we investigated under control of inducible expression system, Cas protein and guide RNA would be expressed and target genomic DNA, resulting in DNA break. For cell survival, supply of 10 plates containing the editing we want can repair the DNA break, resulting in engineered Clostridian cells. The generated CRISPR-Cas systems can achieve efficient and rapid gene disruption and integration in this thesis in about 1 week. And we successfully constructed various recombinant Clostridian with chromosome integrating for producing drugs, including cytokines, antigen proteins, and enzymes. However, we observed that there is a substantial loss of drug production in chromosome integrating compared to recombinant plasmid with high drug production. Besides, as a demonstration using CRISPR-Cas system to efficiently rewrite Clostridian chromosome, we genomic engineer Clostridian cells to produce near-infrared fluorescence for checking of recombinant Clostridian using GI-checked substrate. In this thesis, for engineering Clostridian to produce drug, we started with plasmid engineering and progressed to optimizing chromosome engineering. And finally, we came back full circle to plasmid. But these plasmids are different. They are native in specific genotype of bacteria. Unlike recombinant plasmid, native plasmid is without addition of antibiotic-resistant marker and the replicon from E. coli. The type strain Clostridium butyricum, DSM-10702, has used in this thesis has 2 native plasmids, in which their replicon have been widely used for Clostridian research. Instead of integrating drug production into chromosome, we successfully integrated drug production into these native plasmids. Then we confirmed that using native plasmid can combine advantages with high drug production of recombinant plasmid and stable drug production free from antibiotics of chromosome integrating. As demonstration using native plasmid for gene production, we generate recombinant Clostridium butyricum to stably and highly produce IL-10 to improve its anti-inflammatory function as a probiotic, and cellulase for further industrial applications of using Clostridium eating cellulose. To conclude, firstly, optimized plasmid using Golden Gate assembling facilitates rapid and efficient creation of recombinant Clostridium. Secondly, libraries of gene regulation would tailor drug dosing for using Clostridium as live biotherapeutics. Thirdly, the development of CRISPR-Cas systems expanded possibility for genomic engineering in Clostridium. Finally, the use of native plasmid in Clostridium butyricum provides an antibiotic-free method with increased drug production. These tools were demonstrated in diverse potential therapeutic applications, including cancer therapy like CDPT and CMIT, oral spore vaccines against virus or cancer, and the potential inflammatory bowel treatment. In the future, further validation in vivo or vitro with the recombinant Clostridium in this thesis is required and will unlock new therapeutic possibilities using Clostridium as live biotherapeutics. To the end, I would like to mention one of my favorite propositions from Kerry Mullers, who invented the PCR technique, and his PhD supervisor, John Neelands at University, California, Berkeley. That is, be responsible, you are a scientist. Thanks for your attention.
Thank you for your presentation. The opposition will be opened by Professor Penders, Professor of Metagenomic Epidemiology and Intestinal Microbiology, Maastricht University.
Thank you. Dear candidate, first of all, I would like, of course, to congratulate you with this nice piece of work. I read your thesis with great interest, and you showed a lot of different approaches for genetic engineering of Clostridial species, as also nicely illustrated in your presentation. And of course, besides describing these approaches, you also, and also towards the end of your presentation, discuss the applications for these kind of bioengineered strains. And I particularly was interested with the applications, for instance, for IBD and pathogenic infections. And of course, I would also like to extend my congratulations to your supervision team. But obviously also I have some questions that I would like to address with you. And I would like to start with the things you mentioned towards the end of your presentation, the future applications and the barriers and the need for further validation that you addressed. Because there are some barriers, especially when we talk about the application of strains as probiotics or as oral vaccines. Such as you described in chapter 7 and 8. And especially one of the problems with probiotics, for instance, is that many, if not all, do not persistently colonize in the gastrointestinal tract due to the colonization resistance of the microbiome. But this is, of course, all evidence for non-bioengineered strains. So I was wondering if you have an idea whether these foreign bacterial strains, not being part of our own microbiome, do not persistently colonize, whether that's also applicable for these bioengineered strains. And do you think that has an impact on the efficacy of these strains?
Highly esteemed opponent, thank you for your words. My quick think is reminding me of one report from Clostridium botulicum testing in animal models. They prove that the probiotic is true. It's hard to colonize into the microbiome in the gut, but they do see lots of increase of like the acid produced by Clostridium botulicum in the blood, which means without the colonization, the bacterium somewhere it's already producing something, but in where that we don't know. That's with engineer the therapeutic protein things. We are not target the easy diseases, but rare disease like, because in Clostridium difficile infection in hospital, the microbiota, fecal microbiota transplantation is a promising process or solution to treat these diseases. Maybe we can also think about probiotic strain with engineered therapeutic protein. In somewhere they will put maybe, but I cannot say it, but it depends on the vivo work in the future. Maybe we can see the production of therapeutic protein as the acid producing.
Okay, so basically what you say, for some applications where the microbiome is completely disturbed, like in C. diff infections, this would indeed maybe work better than in other applications. So I was wondering, so for other applications where we still have an intact microbiome, for instance with IBD also, there the microbiome is of course perturbated, but not as much as in C. diff infections. Would there also be other ways to use these bioengineered strains, for instance, towards more precision-based or personalized bioengineered strains? Would that, in the very long future, not thinking about regulations, be a way to go, and how could that be done?
I think that depending on the RNA-seq and DNA-seq to define the species of each one's microbiome from the fecal and feces, then we can personalize the microbiome treatment in the future. And I think it's quite long way to think about this personalized microbiome treatment, but I believe this is the quite interesting aspect to investigate.
Yeah, I think I fully agree with you that it's a very long stretch, but I like that you agree that it might be a possibility in the long future. Maybe to go a little bit more into the details of your work, I would like to move with you to chapter 8, and specifically the part where you worked on the IL-10 producing strain, with the results presented in figure 4 at page 203.
Yep.
So I already mentioned that I have an interest in IBD and the microbiome, but also microbiome-mediated manipulation. Here you used these native cryptic plasmids of a specific Clostridium butyricum strain as a proof of principle for IL-10 producing strains. What you showed is that you were able to successfully and stably secrete IL-10 when you used either integration of the gene into the chromosome or in the native cryptic plasmid PCB102. But when you use the PGG2121 plasmid, you showed, and that's visible in figure 4b, that the production is being lost over successive cultures. But then in the follow-up experiments, you used the supernatant of that strain. And I was wondering, why did you use the strain that loses the IL-10 production over the course of successive cultures? And why not using the strain that was able to stably produce IL-10? Was there a reason for that?
This is a quick process to compare the plasmid, recombinant plasmid and chromosome integrating to lose the antibiotic-resistant plasmid. This is a quick process to see the difference between these three strategies. So that's why we use this strategy. But yeah, that's the reason.
So it's more for the convenience of,
yes,
of the higher
yes,
production.
yeah.
Yeah, I agree. And then what I also noticed is that the culture medium that you used itself already had some immune stimulating effects and also resulted in the proliferation of PBMCs. And if you then look at figure 4e, what you see is that indeed what you describe, the medium itself has some proliferative effects. But when you then use the empty strain, you already see some repression of this proliferation. So even without the IL-10 production, apparently the strain already has an effect. Do you have an explanation for that?
There are some literature review. They tested not only in vitro and vivo work, showing the Clostridium butyricum as a probiotic, well has the repression of the PBMC in FN gamma pathway somewhere to regulate the cell proliferation.
And is it known what is the explanation for that? Could it be the butyrate?
Most of the results think it's because of the acid, the butyric acid produced by the Clostridium butyricum.
Okay. Is there time for one follow-up?
Short.
So, okay, very short. You showed when you looked at the open reading frames of this plasmids that were present in this strain, that one of the open reading frames was encoding a toxin related to, and I have to check, zonulin occludens toxin domain. And you relatively positively framed that, but it is known that this toxin also affects the permeability of the gut. So I was wondering, is this really a strain that will be beneficial, or should we be careful when we move forward to using it as a probiotic?
If I understood correctly, it's talking about the prediction of the genes in the plasmid. Yes, when I blast this gene and find this potential toxin, yeah, it's in one of the plasmid. But this strain we use is a type strain in Clostridium butyricum, has been, since he first discovered it 150 years ago. And in that plasmid, one of the plasmid it's also existing most of probiotic Clostridium botulinum. So that one, I think, it's without of the toxin protein or genes. But the other one is quite a rare plasmid, which would be a concern for using this type genotype bacteria. But if we can think about another probiotic without the toxin, then we can use one of the same strategy to engineer the native plasmid in other bacteria.
I'm pleased with your answer. Thank you. I give the word back to the prorector.
Thank you. The opposition will be continued by Professor Brull, Professor of Molecular Biology and Microbial Food Safety, University of Amsterdam. Thank you for joining via the World Wide Web.
Thank you very much, Mr. Rector. Yes, indeed, I'm sorry not to be in Maastricht today, but this is fortunate to have the opportunity to join in this way. Indeed. Dear candidate, I enjoyed reading your thesis. Very interesting, quite heavy also, lots of pages. But I would like to start discussing a bit, and I would congratulate you indeed with the completion of the thesis, but also with the fact, that's what I will start with the fact that you have propositions. And maybe one of your paranames could read out proposition number 6.
Yes, number 6, right?
Yes, it's number 6.
Yeah, Clostridium species are a promising and versatile vector system for use in cancer therapy. Yes.
Thank you very much. Question is, why are they so promising, these anaerobic bacteria, rather than other anaerobic bacteria? Can you comment on that?
Highly esteemed opponent, thanks for your words. The Clostridium spore via intravenous injection, it has been proved that the Clostridium can specifically colonize the nucleotic region of tumor because of the hypoxia environment inside and the nutrition produced by the nucleotic cells. And compared to the other bacteria, they mostly using the directly inject into the tumor inside instead of intravenous injection.
Thank you.
Yeah.
Very, very pleased with your answer because you mentioned spores, Clostridium spores. And I think that's in fact where I wanted to go to. If I look at your cover, I mean, there's one thing that I don't see, and that's a spore. I think at least, if I'm correct. But on these spores, can you comment on how the spores transform into the vegetative cells and how efficient that transformation has to be, or whether we can gain or should be gaining something from knowing spore germination processes?
Sorry, can you slow down your question? Because
the question is the spores, bacterial spores. What should we know from these spores to study spore germination still?
Most of spore study are in Bacillus subtilis streams, and Clostridium spores. The process it's kind of different from the studies or the knowledge we have, and the common is the spore is less immunogenic components in the surface, which could be advantage for some specific applications.
Yes, definitely. I completely agree. But also maybe the efficiency of spore germination, how well the spores germinate, depends a lot on the compounds that the spores will see. So maybe different tumors provide different compounds, different germinants that the spores can respond to. So that's something to test indeed if you go to in vivo situation to test the germination systems of the spores. So that's a very nice point, and something for the future. I'm very interested in this toolbox that you created, and the question I have there, and then let's go to chapter 4 in detail, is how to use the CRISPR-Cas system, CRISPR-Cas12 system. And you showed in this chapter something about good and bad. What was it again? Pre-CRISPR RNAs. Can you explain to me what generic rules are for good or bad pre-CRISPR RNA structures?
The rationale of designing this pre-guide RNA, we were thinking about maybe we can simultaneously target different sites to simultaneous engineering different genomic sites, and the second one direct repeat region, it's to will be deleted by the processing of Cas12 itself. It's non-process, and it will also cut out of before the terminator on the plasmid. There will be some sequence that we don't need it.
Right.
Then we'll also cut it off, which will help us to more we hypothesize that it will be more target with the.
Okay, I follow. But I have a question on. Let's go to figure 3 on page 90 of your thesis. This figure shows some secondary structure of the pre-CRRNAs, and it shows bad and it shows good. And can you now predict, based on primary structure of the CRRNAs, whether something will be bad or a good version? Is that possible from your experience?
I cannot understand the question. Can you slow down?
Well, page 90. 90.
Yep.
You have good pre-rRNAs in figure 3a. Figure 3a shows good pre-CRRNAs. Figure 3c shows bad, so 3d, bad pre-CRRNAs. And can you explain to me why one is good and the other one is bad?
As you can see, the two loop is recognized by the Cas12 protein. And the first one is the totally correct loop sequence, which will to form a correct complex of Cas12 and guide pre-CRRNA. For the bad one, you see the right loop is correct. It will cut out by the Cas12, but in the front you can see the big loop, which it's wrong. But the prediction of this structure in the system we use the published RNA website, and all of them will have a percentage of this RNA. Some of them will have more because the RNA is not stable in one structure, and this is the most percentage of the guide RNA shows in predict by the website. So which means the bad RNA, they can maybe also in some certain, we don't know, during the experiment may form some good structure of guide RNA structure to connect with Cas12 to target. As we see in the figure E, we can also see bad guide RNA can also achieve somehow in editing efficiency.
Exactly, that's where I was getting. So interaction with the Cas12a, and the prediction of the interaction you have done. I haven't touched upon your website yet, but I will, because that's what I was after, to try to get some generic principles, some overall principles of how interaction works. And I was really very much impressed by this possibility to predict the good the percentage of good or the percentage of bad, if you like. Could you see a possibility that artificial intelligence might help there to predict based on structures that you have identified in future for us? Is that Is that something that you consider also? The use of AI?
This is not my field, but well, of course it's not about AI deep learning recently. So every science can think about AI help, like to open the hand of that scientist to have more interesting or important work to further move on. Yes.
Indeed.
Yeah.
Mr. Rector, is there room for a question still?
No, unfortunately, we have to move to the next opponent.
Okay.
Thank you for your
Thank you very much.
Thank you for your contribution. The opposition will be continued by Dr. Lopez Contreras, a senior scientist in applied biotechnology in a research and development environment affiliated with the University of Wageningen. Thank you for coming to Maastricht.
Thank you for your introduction. Dear candidate, I enjoyed reading your thesis, and I have some questions that I want to discuss with you this morning. Very briefly, the cover picture has already been referred to, but could you explain what are these threads, or can you explain a little bit what we see?
You mean the cover?
Yeah.
From the image we chose from the website, they said it's the Clostridium botulinum on the tissue. It's contamination by the Clostridium botulinum picture.
Okay.
Yeah.
So this is botulinum, and what are these threads?
Well, they are just the color, but
Just the color. Okay. Yeah. Okay. Yeah, I was curious because yeah, it's very nice to see the spores and everything. But this is thank you. I have a question on the page 58 on the chapter 4 on the gene regulation studies. You are studying the different promoters to establish the strength of the promoters in E. coli and several Clostridium strains. And in the figure 2b, we can see the specific activity in E. coli and in Clostridium sporogenes of these promoters. I can see that the specific activity in E. coli is normally a factor between 4 or 5 higher than in the Clostridium case. How do you explain this?
Esteemed opponent, thank you for your words. It's hard to really to say E. coli and the Clostridium sporogenes expression level differences in comparison in these two because the growing condition is different. One is in the shaker with the oxygen, like for E. coli, well growth process in outside of anaerobic region, but Clostridium is collected in the cabinet. And the growth, they are quite similar, like 20 or 25 minutes for one generation.
Yes.
But the nutrition and because the medium we used for the E. coli, it's quite defined, and E. coli is quite easy to grow and culture. But for Clostridium, the medium we used is rich. It's with rich nutrition, but we don't know which is good medium still. Yeah.
Yeah. Okay. Thank you. Yeah. This. Yeah. That brings me to my next question, because you have used a very rich medium that is containing peptone, yeast extract, thioglycolate, and I wonder if you have looked into the medium composition, because this can play a very important role on the activity of the promoters, because you have chosen several promoters from different strains and that are involved in different routes, metabolic routes. Have you considered this during your design of experiments?
I think the problem should be most coming from native plasmid, which is regulated by Clostridium cell itself. So it may be the native plasmid we used may affected by the nutrition of the composition of medium. That's we can, but the other one is we use the consensus plasmid predict by the bioinformatic tools. I think that's one is more like synthetic promoter, which could be not the issue with the changing of the regulated by the nutrition from the medium. But it's true. It's I agree that the medium composition might affect mostly in native plasmid.
Yeah. Okay. Thank you. Also, indeed, when you are comparing the expression of your enzyme of interest, you chose an OD of 1 in both cases for E. coli and for Clostridium. But did you check expression in other growth phase? Did you follow the expression of your protein of interest during growth in these organisms, or you are really focused only on one time point? Because that could be also, of interest for your pattern.
That's good point to test the whole growth phase for the promoter to see like which one is more early phase promoter or late phase promoter. Yeah. But for the gas assay we used is published, and it's a common. People collect this OD1 to have good result for the activity of the enzyme. But for the fluorescent used later, it's more convenient to see the growth during the growth, the fluorescent expression. We can see after the mid phase expository will have less expression level, mostly maybe because of the nutrition and protease produced by the collagen itself. For the gas assay, it's hard to process we can, because after that we can divide the OD to standardize the activity, but the fluorescent reporter we used for the CRISPR-Cas12 system have shown that after around OD1, maybe later, the expression will go down.
Okay, so you can see. Do you see also an effect on sporulation on Clostridia or?
We didn't really look into the process of sporulation, but this is quite important for us to further investigate using the CRISPR system and promoter regulation to deeply understand the genes in collagen genomics, because we use spore as a delivery for the application, but we didn't yet.
Okay, thank you. Also, I have a question on the page 66 on the alternative start codons. It is very typical for Clostridia that they are able to use alternative codons. I mean, that E. coli normally only recognize AUG, but Clostridium or other species, there is a big variety. I think this is very interesting to study because for regulation is really relevant and it's important. But I was wondering, because you have designed new promoters and you have changed the start codons of these promoters, but I think the fact that Clostridium has these alternative promoters, these alternative start codons, there should be a relation with the promoter, or maybe the native promoters where these start codons are there, there should be some kind of relation with it, some kind of function. So did you look at this? Is a certain type of start codon is related to a certain family of enzymes or certain metabolic routes?
Well, we didn't, but I really want to look at this start codon. Well, for me, it's the Pandora box opened by by accident. True, in most, when I look at the genome of the annotation of the Clostridian species genomics, we can see some codon sequence with the start codon like AUU or AUUG start codon, and some of them is quite important enzyme or genes. And if we can use, well, to think about high throughput to compare the genes using non-AUG start codons, maybe we can find some clue of the why they use this AUU to regulate the expression. Maybe AUU is more efficient in Clostridian somehow.
Thank you. In view of the time, I'll give the word back to the chairman. Thank you.
Thank you. The opposition will be continued by Dr. Steen Redeker, Assistant Professor in Synthetic Biology, Maastricht University.
Okay, thank you. Dear candidate, of course, first of all, congratulations with your work. I can see, and I've read it with great enthusiasm, I can see that you did a lot of work. You did a lot of cloning. And I think only memorizing all the different plasmids that you made is already 4 years of work. And I also know that cloning is not always an easy thing, so all the assemblies. So congratulations on that. I have a few questions, of course. Being fourth in a row, it means also that some of my questions are already stolen. But I will start with one of the questions that was in the discussion previously, so now it's still fresh in the memory. So that was about the expression levels. So the figure 2 on page 58, I think. So you have the different promoters, and you compared the activity. And you just said that you standardized the results by dividing it by the OD600, right? Of course, that is done, but can you compare the OD1 for both organisms? Does it mean the same thing if an OD1 of E. coli is at the same?
Like from my eyes, it's different. It's especially even between the Clostridian sporulin and Clostridian butyricum, their colony is different on the plate, and also the median itself is different. So the OD could be well, maybe OD 600 is not the ideal optimal density value to see the really growth of the Clostridian. But the OD 600 is known for bacterium, mostly in E. coli. So that would be a problem.
Because it's scattering, it's not absorption of light. It depends on the size of the bacterium that you are measuring. So how do they compare the size of? Do you know?
Well, I actually don't know, but well, we don't want to really compare the one promoter. Really say like E. coli can produce very high, or 4 times, 5 times compared to Clostridium, because these are two different growth conditions, a different median. Well, the easy way is like we may use E. coli, because E. coli can also grow in anaerobic region cabinet. We can put E. coli in cabinet and without shaking and see what's different, because E. coli need oxygen to have good TCA cycle to produce things. And maybe Clostridium will be better, but we don't know yet.
Yeah, my point was a little bit that because I was also surprised by the factor difference between the activities between the two organisms. Maybe it's just because there's more E. coli than there is Clostridium.
Yeah, the start codon is recognized by complex process by initiate tRNA, and depends on the abundance of tRNA and how stringency of the tRNA recognition process. Let's keep it as a further investigation. We can, because this is quite interesting, because if we have more spare tRNA with low precise recognition process, maybe we can use this start codon as most people like published recently to create a firewall for Clostridium itself too, because every transferred genes will not will translate into a protein, but Clostridium can. That's the idea for the future investigation.
Yeah.
But we don't know why Clostridium can efficiently use non-AUG starter codon.
Yeah.
But it's quite interesting to investigate it.
Okay, then I go back to a more general question about the actual application of this. So basically what you want to do, you want to have the drugs at the right place, right, in a tumor. And you want to have it functional. So for example, you can inject the spores into a patient, and the spores go to a tumor or something else. So you need a specific environment where they can grow, basically. Is there a difference between, let's say, smaller tumors, larger tumors, other areas in the body that have the same conditions, environments, where your Clostridium ends up and produces the drug?
Well, actually, there are some normal hypoxia region for normal tissue, but collagen cannot colonize in that level of hypoxia because big tumor maybe like this need. We don't know which value of hypoxia % of oxygen collagen can really colonize into, and the critical things they need nutrition, and the tumor, necrosis tumor lysis and will expose some nutrition, amino acid and protein for collagen spore to germination. So it's harder to, it depends on the hypoxia environment, the oxygen level inside.
Mm-hmm. Yeah. Okay, and related to this, and this might just be speculation, it's not a question with a right answer or something, but I was at a conference last week where they kind of presented a similar thing, where they target different bacteria in a tumor, but not by a manipulated, let's say, bacteria, but they used a fusion protein to target the protein to the tumor based on the bacteria that are present, like Clostridium, and then that fusion protein had an enzyme that can basically convert a prodrug into a drug. But it's adding the prodrug in the bloodstream instead of having a bacterium that is manipulated go to the tumor. If you know what I mean.
You mean
could that be an alternative option? Because now you have, let's say, a GMO inside your tumor, and in this case you just target your prodrug to the tumor by using the bacteria that already are present there.
The advantage from collagen, well, we have GMO things, which is kind of headache still, but collagen can consistently produce the enzyme and convert the prodrug into drug. For the drug, if I don't understand correctly, you want to directly put the enzyme or drug without bacterial issue. That maybe we need a multiple repeat injection of doses. This is the big difference, I think.
Yeah. Okay, one short question. You talk about codon optimization a few times. So for protein expression, you can codon optimize a protein for that expression. But you prepared vectors for direct cloning, but you have to do that every time again, right? Because it's dependent on the enzyme or protein that you want to produce. What is the optimal codon usage? Well, actually in IDT, for the company, they have a web tool to codon optimize your genes from if you give the amino acid sequencing or gene sequencing, you can choose the organism. There is option collagen. Well, the well studies Clostridium acetobacterium. They use that as a template, as an option. You can use this to optimize your genes. But the more additional way, we will also look at the repeat sequence, because we don't want a high multiple repeat DNA. Maybe will affect the DNA stability. So we can
well, there is a web tool to optimize. It's quite easy to, then we just click and order the optimize the genomic genes. Yeah.
Okay, thank you.
Thank you. The opposition will be continued by Dr. van der Beuken, Assistant Professor of Translational Genomics, Maastricht University.
Thank you, Prorector. Dear candidate, I also want to congratulate you with this very nice thesis and the impressive amount of work that you did. I also want to congratulate the promotion team with this great performance. I think you generated a lot of valuable tools for the research community that a lot of people will like to use in the future. I've got 6 pages of questions, but I don't have the time to ask them all. Luckily, many of the questions have already been answered. But let me start with, so I'm mostly focused on all your efforts that you did to optimize gene expression, using different promoters, testing different UTRs, the alternative start codons that was already discussed. And I understand that you want to sort of you strive towards a system that has low expression in E. coli, but high expression in Clostridium, right? But maybe a very naive question to start. If you want to clone lethal genes, it's understandable that you want them not to be expressed in E. coli, but would they not also kill the Clostridium cells?
The toxicity of protein gene expressed, it's, well, will have lots of reasons. One is transcription, mRNA level, and also the protein itself toxicity. And in the promoter library, we use high copy plasmid. Then we also met lots of cloning issue because of the copy number is too high and produce lots of mRNA level and also the protein. But for the secretion protein we produced, the toxicity we hypothesize is because of the signal peptides from the Clostridium is also recognized by the E. coli. And using the signal 6, well, also a web tool created by DTU, the Denmark Technology University, they, if you choose different bacteria or setting with gram-positive and gram-negative, they pick the same signal peptide sequence, which meaning that all cloned secret genes we cloned into plasmid will also secrete out to E. coli. Well, the protein will stay in the between because of cell wall gram-negative bacteria. The protein will not further go out to the, well, it's not efficient go out to the culture or medium, and it depends on the soluble dissolving of the protein and also the expression level. The protein might stay in the envelope of the E. coli, then maybe causes cell fragile and toxicity. But for Clostridium, the protein will secrete out to the cell membrane, and using the diffusion, the protein will go out freely to the medium, which is, well, which might be the explanation for the toxicity we met. But the toxicity reason it's hard to say, because also some research, they express the metabolic pathway genes in E. coli will also cause the cell toxicity because it changed the metabolic pathway in E. coli will also kill in the E. coli. They are not happy, then they will destroy your genes. And yes, that's why I think during the conference, I met people in Germany, well, a PhD student, he has a headache cloning 1 gene from Clostridium metabolic pathway. It's hard to clone this enzyme in E. coli because it's totally different pathway in anaerobic bacteria. Then you will have the metabolic pathway or, well, toxic product produced by the enzyme. Then
you may interrupt.
Yeah, I was just thinking about okay, thank you for your question, but yeah. I was very interested in chapter 6, and there you sort of compare the expression of plasmid-based transgenes versus the integration of these genes. I think that's a very elegant way to integrate these into the genome of Clostridium. But then you also show that the plasmid base have a far superior expression level compared to the integration. And I was thinking, like, can you think of other ways how you can maybe increase the expression of this integration-based system in terms of, would it be possible to maybe change the integration side or do something? Do you have any ideas about that?
In this thesis, we express the genes, we clone the genes to integrate the gene with two terminator outside of gene expression cassette. And this is the quite safe way. I think it's the it's quite a safe way to protect the genes in the genome outside, because the promoter inside the expression cassette will not affect the downstream expression level. But if you leave this out and put somewhere integration, then the expression level of native in the genome will also improve the expression of. Yeah.
You may finalize your sentence, but you can also stop if you prefer.
Also, yeah. And also you can integrate multiple copy in the genome. And there is a publication saying that if you integrate your expression cassette in replicon of the chromosome, it will somehow during the replication of cell, the copy number of your integration will be much higher than you will maybe the production of gene will have may higher there.
Okay, thank you. I'm happy with your answer.
Thank you. Yanchao Zhang, the time appointed for defending your thesis has passed. The committee will now withdraw and to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. bye now, don't waste all your time. Cause I'll go the extra mile.
Yanchao Zhang, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. Professor Lambert is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Yanchao Zhang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificates signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. I will now give the word to the co-promoter, Dr. Jan Thijs.
Yes, thank you. So, Yanchao, Dr. Zhang, as we from now on, Dr. Yanchao Zhang, as we from now on can say congratulations to you, to your family, your girlfriend, and to everyone who has been supporting you, I think, throughout this journey. You did it. You made it. And actually, it is not a surprise at all. Because from actually the very first letter, so we received also at that time lots of applications from CSC students, yours somehow stood out, and it was very clear from the very first interviews that we did online that you would be the candidate of our choice and that you would be an excellent candidate. And that is how it turned out. Not that it has been very easy or straightforward. Initially, I think the language was maybe also a barrier, but as in so many other areas in the past 4 years, you showed also there a steep learning curve. And maybe not only the language, also the culture, because you end up from a 15 million inhabitant city in China into Maastricht, which has a little bit less to start with. And also, you have to get used to the, let's say, direct Dutch way of handling. And I can tell from experience that is not always so easy. So, but also there, you did well. Also, you have to get confident or a bit used to the habits, the food, and so on. But you found your way. I think firstly connected to predominantly also your Chinese fellow students, but gradually also to the others in the lab. And from the start, there has been, and it has came across in the discussion here also, and also in what we said behind the doors there, and that is your dedication to the work. You are always very dedicated to the lab work from the start. The lab work, the experiments, long days, working in weekends and so on, problem solving, optimizing. You were always busy, and if it was not in the lab, you were reading papers, getting ideas, and so on. I'm not mentioning you did it yourself. It was nice. You sort of inserted into your defense during your PhD the headache that you got from all the let's say, regulations and so on. But even so, or despite all these, you sort of got going. And you were also present when we had the activities outside the lab, team buildings, things outside the lab. And actually there, some kind of the first surprise came for me. Because, of course, when we are having these bottles and so on, there's some beer, there's some wine and so on. And then, okay, Yanchao was always amongst the first. I always remember, or also remember the team building that we had in the winery. And of course, yeah, if there was drinks involved, Yanchao was there, which is okay. We're young students, like it's good outside of work. But that was not a surprise. The surprise came actually a little bit later when you went to China and you came back. And then you brought, like, really this, some kind of, what to me is like the closest to pure alcohol sold as like a drink. And showing again your dedication, you also told me that it was like fermented by Clostridium or something. I think I'm a bit, or try to be an ambassador for using Clostridium in medical applications and so on, but this, I don't know exactly where this fits. And I've been wondering afterwards, like, did we have at the time like a conflict? Was there something that was not going well? Because I'm sure this type of gift, this type of alcohol you can use also for someone, but you don't like them so much. But okay, I don't, and I'm actually sure that that is not the case. But all this and all what has been told actually shows that you developed actually from, in my opinion, like rather shy, introvert person at the start to a mature PhD student. And also a student with an opinion, not only when it came to research and new ideas, but also, for instance, you brought forward all the issues related to your position as a CSC student and the limitations that came with this. And I must say, and I think you can be proud, you and some others together with you, by hearing your voice. I think we were within the department one of the first to go level higher and say, like, okay, this is something that we cannot, this is something really that should change. And I'm very happy that actually this week it has been communicated by the university that it will change, or that if CSC students come, they need to have the same rights, they need to have the same benefits and so on. And of course, for you, this is a bit too late. It came a bit, you will not really benefit from it, but it's definitely thanks to people like you, like showing your voice, that has changed. And you can be very proud of it, and we are very happy that you did so. And from this being shy and being always into your own work, what I've also seen, and which is probably the most important, you turned into like a real team player in helping others, sharing your protocols, sharing your expertise and so on. And we are really, really very grateful for that. It has been said here during the defense, you leave a lot of tools for us and for others that we can actually use. And all your work has by any means been unique. And so thank you very much for that. And so today we are celebrating. We are celebrating, of course, the fact that you're here, not only your output, your papers, your patent that is also there, but maybe and especially your creativity and the passion that have fueled your journey. And those who know you a tiny little bit, they know that this has been soundtracked, your entire, let's say, procedure, by like this, some kind of artist, I think you might know her, Taylor Swift. And I'll show you. So in fact, only retrospectively, but then I was thinking when I was making this, actually, maybe it was already from the start that she was there, although we were at that point not aware. And so because, and I looked it up, I'll try to sort of prove my point here, but now I really have to read because otherwise it will not work. So throughout the years, I realized that Taylor's music has become more than just a background for you. Not only did Yanchao attend, I think twice, the New Eras tour of Taylor Swift. Personally, I've been looking for tickets and trying to get tickets, not possible. You got 2 times there. It's just, again, something about, I think, your dedication, and of course also because you might be a bigger fan than I am, but okay. Just as, so as a devoted fan, you know that success often requires resilience and creativity. Themes, of course, that resonate in Taylor's music and the world of academia. And the message also at the heart of Taylor's song, I did look it up, really, shake it off. Shake it off in the face of criticism and to never go out of style when you are pursuing your own dreams. You have shown us how to keep moving forward also when the road gets tough, and that it was, of course, for sure. So in your own way, you have taken on new eras, facing every challenge with the same determination that Taylor talks about in The Man, becoming fearless in pursuit of your goals, just as Taylor encouraged us all to be. So there are probably moments when you might have wondered if you would ever reach this point, like today. Perhaps in the midst of late-night writing, which you certainly did, you found yourself humming All Too Well. Because we all know the PhD journey is full of those long hours and painstaking edits. But, like Taylor, you also persisted. And as she says in the final, in Long Life, I had the time of my life, fighting dragons with you. You have undoubtedly also fought many dragons along the way. And today, you stand here victorious, ready for a next step in Toronto as a postdoc. Of course, good luck on entering this new era. I'm sure you will do well. And I would like to end, just because I think it is, it makes sense and is applicable, with a short poem of a Australian young poet, Eric Hansen. And here it is. There is freedom waiting for you on the breezes of the sky. And you may ask, what if I fall? Oh, but my darling, what if you fly? Congratulations, it's well deserved. Thank you.
Dear Dr. Zhang, also on behalf of Maastricht University and the whole community, I congratulate you with the degree you have acquired. And I wish you a lot of success in Canada with your future work. Unfortunately, Taylor Swift was not available to congratulate you, but I'm sure that Professor Brull will take her place.
That's quite a challenge, Mr. Rector. Thank you very much. No, indeed, dear candidate. Well, now I should say, dear Dr. Zhang. Of course, very well done. I was really pleased with reading the thesis, also with a lot of your discussion points that we had. And I would just say a lot of wishing you a lot of good fortune in the future in Canada as a postdoc. And there's a famous Dutch microbiologist who lived a long time ago, called Beijerinck, Martinus Willem Beijerinck. He's in Delft. He was in Delft. And there's a saying that he also always used, and that says, fortunate are those that now start. And I think you are indeed fortunate, and we are fortunate that you have started and are going on. So keep up the good spirit, with or without Taylor Swift. I also like the songs, by the way. So have a nice time in Canada and have a nice time today. Thank you.
Thank you. With this, I close this academic ceremony. We will stay here for a few minutes to make some pictures, and we meet each other in the after. Thank you.